chemical basis of life and not cellular basis. We will keep cellular basis aside until it finishes. Let's keep cellular basis portion one side, and we will try the chemical basis of life. 50 MCQs. If you are okay, tell me. You have to get ready. So how far we have worked on this lesson? The endoplasmic reticulum is over.
Yes, sir.
Lysosome.
Done.
Peroxisome.
Done, sir.
Mitochondria.
Done, sir.
Chloroplast.
Done.
Cytoskeleton.
Done, sir.
Cilia flagella.
We have to start that, sir.
That's so. If you take cilia and flagella, cilia and flagella can be noticed as external extensions. Okay, cilia and flagella can be noticed. Cilia and flagella can be noticed. External extensions. Okay, some hair-like projections can be noticed from the cell surface, and those hair-like projections are the one called by the name cilia and flagella. In the case of cilia and flagella, if you see this picture, the blue color material surrounding the cilia and flagella is the what is it? Plasma membrane. It is surrounded by the plasma membrane. Okay. Within the plasma membrane, can you see these tubular structures? Can you see these tubular structures? The orange color one? Yeah. They are microtubules. They are microtubules. Right? When two microtubules stay like this, attached to each other, it is called microtubule doublet. What is the name? Microtubule doublet. Okay, if you count the microtubule doublets, how many microtubule doublets are there?
9.
9. If you look at the middle, in the middle, microtubule 2 are there, but they are not staying as a doublet. Understood?
Yes, sir.
And they are called what kind of microtubules?
Central
microtubule. Outer microtubules are occurring in the form of doublets. Central microtubules are separate. They are not occurring as doublets. That's why we refer this as 9+2 arrangement. That word is already familiar to you. Correct? That word is already familiar to you. If you take this middle diagram, as usual, this is okay, it is the TEM diagram of cross section of cilia O, what is it? Flagella. Mark it down. TEM, transmission electron microscope diagram of cross section of, what is it?
cilia or flagella.
Cilia or flagella. In exam paper, they might project this one to you and ask what it is. Then the answer you provide should be perfect. Understood? It should be a perfect answer. Some children, they write cilia, flagella. Will they give you marks for that?
No.
No. Perfect answering. Everyone is answering the exam paper. Some only perfectly answering the exam paper. Correct? Everyone is answering the exam paper. Some only perfectly answering the exam paper. They are becoming the best performer. Okay, we cannot be 1 of 100. We should be the best. Here, the opportunity, the future is waiting only for the best. So we have to try ourselves to be the best. Clear?
Yes.
Yes. So have you marked it? Right. If you take the cilia or flagella, in the bottom, they are anchored to a unit called, what is the name of the unit?
Basal body.
Basal body. Some places they call it basal granule also, but basal body, that is well sufficient to know. So basal body. If you look at the basal body, it has a different microtubule arrangement. You can see here, microtubules are arranged not in the form of doublets, they are in the form of triplets. Can you see?
Yes.
Yeah. So how many microtubule triplets are here?
9.
9. Do you notice any microtubule in the middle?
No.
No. This is described as 9+0 arrangement. So what kind of arrangement is found in the cilia?
9+2 arrangement.
9+2 arrangement. There are 9 mean 9 doublets in the peripheral. Agree?
Yeah. Yes.
Here in the basal body, what kind of arrangement there?
9+0 arrangement.
9+0 arrangement. Here 9 indicates 9
Triplets.
triplets of microtubules. Here also the central diagram shows you the cross section of the basal body. As I told you, it is the TEM diagram of cross section of what is it? Basal body. So in the tube, you include it TEM diagram of. In exam paper, if they ask you, that's the way to answer it. TEM diagram of cross section of basal body.
Done, sir.
Completed?
Yes, sir.
Right. So, if you take cilia and flagella in a cell, cilia will be very large in number. Cilia will be very large in number. Thousands of cilia will be there. In one cell, thousands of cilia will be there. But if you take flagella, flagella will be few there. 1, 2, 3, 4, okay, 5, 6, like few will be there. Can't expect thousands of cilia or flagella. Understood?
Yes, sir.
Right. And cilia is short and flagella is long. If you take their functions, what is the first function given to you? What is the first function given to you?
Act as locomotor appendages.
Locomotor. Act as locomotor structure. Okay. Here you can notice can you see a euglena moving?
Yes, sir.
Yes, sir.
Can you see its flagella?
Yeah.
Yeah. How many flagella euglena has?
2 flagella.
2 flagella. Right. Similarly, so tell me one organism that uses flagella as locomotor structure.
Euglena.
Euglena. Similarly, paramecium. You know paramecium. Familiar word. If you take paramecium, the word is very familiar. Can you see the paramecium here?
Yes, sir.
Can
Yes,
you
sir.
see its ciliary bead? You can't see the paramecium?
Can see, sir.
We can.
Amna, can you?
Yes, sir, I can.
Okay. Can you see the large number of cilia beating?
Yes, sir.
So that is paramecium. Okay, then act as a locomotor appendage. Okay, flagella as a locomotor appendage. For whom?
Euglena.
Euglena. Correct. Cilia as a locomotor appendage. For whom?
Paramecium.
Paramecium. Other two, now further, okay, further, now animal sperm, animal sperm use, what is it? Flagella as a locomotor appendage. Further, cycas sperm. Do you know the cycas plant?
Yes.
Yes. If you take, okay, not cycas sperm, okay, plant, non-flowering plant sperm, non-flowering plant sperm. Cycas sperm coming here. Okay, so non-flowering plant sperm. There are many non-flowering plants you studied in your grade 10, in the grade 10, yes, grade 10. Here you will be getting lot will be coming. Lots of details are there, but not coming in the unit number 2. It is coming to you in the unit number 4. And little coming in the unit number 3 also. Okay. So, who are the structures using flagella as locomotor appendage?
Euglena, animal sperm, non-flowering plant sperm.
Non-flowering plant sperm. Cilia as locomotor appendage.
Paramecium. Cycas.
Paramecium and cycas plants sperm. Remember, cycas is a non-flowering plant, but specially it is locomoting through cilia. Other non-flowering plants are using flagella. But anyway, that much detail is at this moment not necessary. You just take this one down. For our MCQ, it will be useful.
Done, sir.
Completed?
Yes, sir.
Right. What is the second one you are pointed? Moving fluid over the surface of the tissue. That is done by whom? That is done by whom?
Cilia.
Cilia. Example, our which tract? Which tract? Respiratory tract. Now the phlegm-like material, correct? From inside are thrown out by the movement of whom?
Cilia.
Cilia.
Similarly, cilia lining the oviduct. Give me 1 minute. Okay, one thing. Last leg. Okay. Cilia lining the oviduct. Oviduct means it is another word given for fallopian tube. Do you know the fallopian tube? Do you know the fallopian tube?
Yes, sir.
Yes, that is also called by the name. What is it? Oviduct. Inside, okay, from the ovary, an egg will be released. Unlike sperm, egg has no self movement. Sperm can move by itself. Okay, sperm can move. Okay, like this, it can move using the flagella. Sperm can move, but that way, if you ask, can the ovum move? In ovum, egg cannot move. So to make it move, what is available is cilia. In the wall of the fallopian tube, there will be cilia. The cilia will beat and make the ovum to move through the fallopian tube. So, if you take the second one and the third one, second one and third one, these two, if you consider, those two are not related to locomotion. Understood? These two are not related to, what is it? Locomotion. And they are done by whom? They are done by whom? Cilia. That means you can make a decision. Flagella involves only in locomotion. Understood? Flagella involves only in
Locomotion.
locomotion. It is the cilia performs locomotion function and non-locomotion movements also. If you take locomotion, it's a movement, fluid moving, egg is moving, all are movement related function, but all the movements are not locomotion. Clear?
Yes, sir.
Good. So you can mark this point, then we can move further. Have you marked it, Putta?
Yes, sir.
Yes. So we are coming to the next heading called, what is it? What is the heading?
Centrioles.
Centrioles. Look at this diagram. This diagram is an animal cell. What kind of cell?
Animal cell.
An animal cell. So specifically, centrioles are situated or found in animal cells closer to the nucleus. Understood?
Yes.
Yes. So mark the animal cell. Mark the animal cell. Okay. Okay.
Okay, sir.
Right. If you take here, this is a centriole. This is a centriole. In one animal cell, you can notice how many centrioles. How many centrioles you can notice?
2.
2 centrioles. Same time, one centriole. Now see, this centriole's axis is located like this. This centriole's axis is located like this, correct?
Yes, sir.
Yeah, the two axes are perpendicular to each other. Understood?
Yeah, sir.
Two axes are perpendicular to each other. In the cytoplasm, the region where you see these centrioles, the region where you see these centrioles is known as centrosome. Where? What is the name given to that region?
Centrosome.
Centrosome. So in one centrosome, how many centrioles are present?
2.
2.
2. How they are oriented?
Perpendicular to each
One
other.
perpendicular to the other. Clear?
Clear.
Yes.
Right. In the centriole, you will see the same arrangement of basal body. What is the arrangement of basal body?
Triplet.
What is the arrangement of microtubules in the basal body, Putta?
Triplet.
9 plus 0.
9 plus 0. Correct? Yes or no?
Yeah.
That 9 plus 0, 9 triplets in the peripheral, no microtubules in the center. The same one can be noticed. Here the picture is not so clear. Okay, the picture is not so clear, but that is the idea. This is TEM diagrams, no, so they will not be very clear. Understood?
Yes.
Right. So
Yes.
that's why I showed you this. You can imagine the same arrangement. 9 plus this kind of a diagram given mean it can be the cross section of basal body, it can be the cross section of centriole because both are looking alike. Okay, Are you clear?
Clear.
Yeah, sir.
Right. During now in your O levels, you studied cell division, correct? Did you study cell division?
Yeah, sir.
Yes.
Yes. Mitosis, meiosis, like words you studied, correct?
Yeah.
In the
Yeah.
A level, you will be studying a lot about it. Okay. In that one, I just choose these words. You don't need to take those words to the head at the moment, but that is syllabus. Here you can see in this picture, okay, there are, can you see there are centrioles here?
Yes. Sir.
The thread-like structures coming from the centrioles are called spindle fibers. What are these?
Spindle fiber.
Spindle fibers. They are made of microtubules. They are made of microtubules. They are here to separate the chromosomes. What is the use of these spindle fibers?
To separate the chromosomes.
To separate the chromosomes. So here, to make these spindle fibers, who is responsible?
Centrioles.
Centrioles responsible, or you can say centrosome is responsible. Understood?
Yes. Sir.
Yes. So it is that function here you are mentioned. See, what do they produce? Read this one.
Produce ester and spindle in cell division.
Ester, read it. Produce ester
And spindle in cell division.
and spindle. Okay, you might ask what is the ester? Already in the picture I showed you, it is there, but I will show an enlarged picture to you. Mitosis, metaphase. And the metaphase is easy to see, that one. That's why. I'll take this. Can you see this picture?
Yes.
Right. In this one, these small microtubules are there, no, the star-like.
Yes. Sir.
They are the one called by the name esters. Understood?
Understood.
The long growing ones are called by the name
Spindles.
spindle. Spindles are made of microtubules. Ester is also made of microtubule. Anyway, they are involving in the cell division, mainly in the animal cells. Right. So see whether you are clear on these components. Centriole is made up of cylindrically arranged microtubules, which are non-membrane bounded subcellular components present only in animal cells. So centrioles can be noticed only in animal cells. They are not membrane bounded. They are cylindrical. They have 9+2 arrangement. 9 sets of triplets are in the surrounding. Nothing mean no microtubules in the middle. A pair of centrioles are arranged perpendicular to each other in the region called centrosome near to the nucleus. The function it is making ester, making spindle during cell division. What do you say?
Yes.
Are you clear?
Yes. Sir.
Yes. So now we are coming to the heading called, what is it? What is the name? Central vacuole. Here, if you take central vacuole, what type of cell is shown to you here?
Answer.
Plant cell.
Plant.
Plant cell is shown to you here. So what we can understand there is, okay, central vacuole is found in the, what is it?
Plant.
Plant cell. If you take the central vacuole will be bordered by a membrane. Central vacuole will be bordered by a, what is it? Membrane. And this membrane, you have, okay, take a color pen or something and little darkly draw it. This membrane is called tonoplast. Sometimes it would be a familiar word to you from your lower grades. Tonoplast. Inside the tonoplast, what is present is called cell sap. Understood? Externally surrounded by a, what is it?
Tonoplast.
Tonoplast. Okay, middle content is
Cell.
cell sap. Right. In the cell sap, there will be water, there will be inorganic ions, there will be sugars, there will be pigments, like that. Several components will be there. Understood?
Yes.
Good. Please mark it down. Have you marked it?
Yeah,
Yes.
sir.
Right. Further, if you take this central vacuole, right? When it collects water, now I'm just drawing a simple diagram. Say this is a plant cell. So this is same type of a plant cell. One cell, it is inside pure water. Pure water. It is inside pure water. Another one, it is inside salt water. Left side one is inside, what is it?
Water.
Pure
Pure.
water. Right side one is inside salt water. When it is in the pure water, water will go into the cell. When water goes into the cell, vacuole will collect the water and will become, what is it? A larger vacuole. Understood? Understood. Okay. When you take the right side one, it is going to lose water. In that case, the vacuole will be, what is it? Shrinking like this. Understood?
Yeah.
Okay.
Yes.
Not only the vacuole, even its cytoplasm will be shrinking like this. This kind of cells are called turgid cell. What kind of cell?
Turgid.
Turgid. Turgid mean swollen, വീങ്ങിനത്. Understood? Okay. This is called plasmolized cell. Plasmolized means shrunk, സുറുങ്ങിനത്. Clear? Are you clear? Right? Now, do you have a small home garden at home? Do you have? Yeah. So, if you don't water the plants, for a couple of days, what will happen? What will happen? You will say, this will happen, correct? Can you see?
Yes.
Can you see? What is happening in the left side cell? What kind of cell is the left side cell?
Turgid cell.
It is a turgid cell. How the plant is looking? Is it fresh?
Fresh.
Yes. Is it straight?
Yes.
Yes. Has it wilted?
No.
No. What type of cell is on the right side? What type of cell is on the right side?
Shrinking.
Okay, what is that word? Practice that word.
Plasmolized.
Plasmolized. So it is a plasmolized cell. So what has happened to the plant? It has wilted. Correct?
Correct.
Yes or no?
Yes.
Yeah. So this is one role play of central vacuole in a plant, making the cell, the plant to stay erect, steady, fresh, is done by central, what is it? Vacuole. Another idea there. After that, we will go for the ideas further. You know, there are, what is it? Do you know this word?
Stomata. Yes.
Stomata. So, do you know stomata can open and close? Do you know?
Yes, sir.
Yeah. Here, look at the left and right. Left side stomata is open. What they have marked here in the bracket,
Swollen.
that means the guard cells are in turgid state by intaking water. Understood?
Yes.
Okay. Here, what kind of state?
Shrunken.
That means
Plasmolized.
plasmolized. What has happened to the stomata? Closed or open?
Closed.
Closed. So stomata's opening, stomata's closing depends on whether the guard cell, central vacuole, collect water or loses water. Clear?
Yes, sir.
You clear? One more example with one more idea. I will go to that. You know the touch me not plant. Touch me not plant. Mimosa plant. Correct?
Yes, sir.
Okay. Why it is called touch me not? If you touch it, it will fold like this. Correct? Yes or no? If you take this leaf, it has been already touched. Because of touch, it has lost the water. Because it has lost the water, it has shrunk. So leaves have folded. Correct? Why this is an open leaf? It is not touched. Its cells are turgid. It is keeping open. Understood?
Yes, sir.
This way, turgidity related movements are called by the name. What is the name?
Turgor movement.
Turgor movement. Stomata's movement is also turgor movement. Touch me not plant's movement is also
Turgor movement.
turgor movement. Are you clear?
Clear.
Yes. So, with this knowledge, let's go in. Here they say, central vacuole is a large structure bound by tonoplast, filled with cell sap in the, what is it? Plant cells. In the cell sap, water will be there, ions will be there, some water soluble pigments will be there, even organic compounds will be there, even photosynthetic pigment, sorry, the water soluble pigment is an organic pigment. We will come to this. So what is the function? What is the function? Read the first function.
Stores water and other materials such as sugars, ions, and pigments.
So do you agree cell sap contains sugar?
Yes, sir.
Yes. Right. It is storage function. What is the second function?
Maintains water balance of the cell. Osmoregulation.
Osmoregulation. when too much of, now if you take a normal man's life, when he earns money more than what he wants, what he will do? Say you are earning money more than what you want. What will you do? What will he do?
Spend.
No,
Save money.
he will save the money, correct?
Yes, sir.
Yes. When his income lowers, what he will do?
He will use it.
He will use that saved money, correct?
Yes.
Similarly, when a cell gets more water, that extra water will be saved in the central vacuole. Understood?
Yes, sir.
When
Yes.
the cell becomes insufficient in water, from the vacuole, it will collect the water. That is what they say, it is maintaining water balance. Are you clear?
Yes, sir.
Right.
Clear.
Giving, third one, gives turgidity and support to the cell. That's what I explained you. When it gets the water, it becomes turgid one, it is staying straight, correct?
Yes.
Yeah. Last one, it is producing colors. It is producing colors. In the plants, you will see many colors, but all are not those pigments. Do you know coleus? Do you know coleus plant?
No, sir.
No, you haven't seen this plant. You haven't seen this plant. Tell me.
Scenes.
Seen. Can you see a pinkish red color, a purple color there?
Yes, sir.
That pinkish purple color or red color is due to the pigment called anthocyanin. What is the pigment?
Anthocyanin.
Anthocyanin. It is stored in the cell sap. Staying in the cell sap, this color coming. Understood? Understood?
Yes, sir.
Yes. Now, can you understand producing color in some plants with sap pigments? Why it is called sap pigment? Why it is called sap pigment? It is the pigment present in the cell. What is it? Sap. Because it is present in the cell sap, we call it what kind of pigment?
Pigment.
Sap pigment. Are you clear these four functions?
Yes, sir.
Yes, sir.
Okay. These four functions, if you take these four functions, if you consider these four functions, okay, these four functions are done by the home. Central, what is it?
Vacuole.
Vacuum. Understood? The last one function given to you now. What is the function given?
Helps in digestion.
That is not done by our central vacuum. It is done by whom?
Peripheral vacuole.
Which vacuole? Which
Food vacuole.
vacuole? Which vacuole? I have highlighted here.
Food vacuole.
Food
Vacuole.
vacuole. It is the food vacuole performing which work? Which
Digestion.
work? digestion. Read these two words. What are the two words here?
Food vacuole, contractile
Food
vacuole.
vacuole and what is it? Read the
Contractile
other
vacuole.
one. What is it? Contractile vacuole. Contractile vacuole performs osmoregulation in Euglena, Paramecium, amoeba. They are only 3 in our syllabus. They do osmoregulation not in the plant. In whom they do the osmoregulation?
Microorganism.
Not all the microorganisms, no puta. So you have to specifically tell which microorganism. Which microorganisms?
Bacteria.
நான் தான் எழுதிதானேமா இருக்கேன். நீங்க ஏம்மா புதுசா கொண்டு போடுறீங்க? Can't you see my screen? Arima.
Can, sir.
Amna, can't you see my screen?
Cancer.
Then what I have written for osmoregulation?
Euglena, Paramecium, and amoeba.
அப்ப நீ ஏமா குழந்தை bacterians? என்ன அநியாயம்? Understood?
Yes,
Amna?
Yeah.
Bacteria can't have contractile vacuole because vacuole is membrane bounded. Vacuole is, what is it?
membrane bound.
Membrane bounded. Can there be membrane bounded organelles present in eukaryotic organisms, prokaryotic organisms? Can bacteria have vacuole? Mitochondria, chloroplast? Can there be?
No.
No. Right. Food vacuoles you can notice in Paramecium. Okay. Food vacuole, you can see them in amoeba, Paramecium, even Euglena, you can choose. Okay. It is the food they intake. The food they intake, they do digestion. So food vacuoles, so contractile vacuoles, they are not found in the plant cells. They are found in certain microorganisms. Amoeba, Paramecium like microorganisms have food vacuole. Okay, Euglena, Paramecium, amoeba like microorganisms have contractile vacuoles. Are you clear?
Clear, sir.
Right.
Yes, sir.
Please include these points there.
Done, sir.
Have you completed?
Yes.
Right. So please read through the central vacuole and tell me whether you are clear.
Done, sir.
Right. So with the central vacuole, it is over subcellular component. We have completed the, what is it?
Subcellular component.
Subcellular components. Now we have to speak about whom?
Extracellular component.
Extracellular component. In the whole extracellular components, you check and tell me what are the extracellular components given. One extracellular component,
Cell wall.
cell wall, correct?
Cell junction.
Cell wall. Second one, cell
Junction.
junction. Third one,
Plasmodesmata.
no, the master heading. And that is the last heading. What is it?
Matrix of animal cells.
It is called extra. நம்ம pocket எல்லாம் எதுவும் போடக்கூடாது மகள். We have to tell what is there. If you write in exam paper, matrix of animal cell collapse. Understood? What it is there, you have to pick. What is the short form of this? Check and tell me.
ECM.
ECM. Okay, they are standard abbreviations. In exam paper, the examiner can directly use it. Which one of the following is correct regarding ECM? Like that, he can put a question to you. Understood?
Yes, sir.
Yes.
First of all, so are you clear? You are going to study regarding whom and whom and whom?
Cell wall, cell junction.
Cell wall, cell junction, and extracellular matrix, shortly called ECM. Okay. So let's go for first material. What is it?
Cell wall.
Cell wall. If you take cell wall, this idea, I am recalling this. Plant cells have, what is it? Cell wall. In that one, the cell wall will be made of cellulose, hemicellulose, pectin, lignin, or suberin like materials. What are the materials making the plant cell wall?
Cellulose. Hemicellulose.
Pectin.
Pectin.
Lignin.
Lignin.
Lignin. Lignin, suberin, only one is possible. That's why lignin, or what is it?
Suberin.
Right.
Suberin.
Second one, it is algal cells. Alga. In alga also, who will be there?
Cellulose.
Cellulose will be there. In alga also, who will be there?
Pectin.
Pectin will be there. Some of them additionally have agar. Some of them additionally have alginic acid. Some of them additionally have silica. Like certain other options are there in the alga, but cellulose pectin will be there. Okay. Third, it is bacteria. If you take bacteria, in bacteria, cell wall contains the material called, what is it? What is the material? Peptidoglycan. And if you take Archaebacteria, another type of bacteria, It's a different domain. In this one, they have polysaccharides and protein. Okay, and in the fungus, there will be the cell wall made of, what is it? Chitin.
Chitin.
So like this, okay, like this, there are many organisms having cell wall. Among these cell wall, bacteria, archaebacteria, fungus-like organisms, cell walls are okay, very flexible cell walls. What kind of cell walls?
Flexible cell wall.
Flexible cell walls, and they are thin cell walls. Thin cell wall. But if you take plant cell wall, algal cell walls, actually compared to plant cell wall, algal cell walls are thin, but anyway, their cell walls are, what is it? Rigid cell walls, and comparatively they are, what is it? What kind of cell walls? Thick cell walls. So what are the organisms having cell wall? What are the organisms having cell wall? Answer me. Answer me. What are the
what was the question?
organisms having cell wall? What are the organisms? Tell me.
Plants,
Plants.
algae,
Algae
Algae.
is L-A-L. L is missing. Alga. Third one.
bacteria,
Hmm.
archaebacteria,
Hmm.
fungus.
Okay. What are the organisms having rigid, thick cell walls?
Plants and algae.
Plants and algae. Flexible, thin cell walls.
Bacteria, archaebacteria, and fungi.
Fungi. Are you clear?
Yes.
Right. Please take a white sheet and mark these details. Have you marked it?
Yes, sir.
Yes. In this lesson, we are given lots of details regarding plant cell wall. If you take plant cell, now we are gradually improving this. If you take a plant cell, say this is a, what is it? Plant cell. The plant cell is here covered by plasma, what is it?
Membrane.
Plasma membrane. Out of this plasma membrane only, who should be secreted? Cell wall should be secreted. Understood?
Yes, sir.
Right. In the beginning, one cell wall will be secreted. I will use a different color for that, for you to easily identify. One cell wall will be secreted out like this. This cell wall is called primary cell wall. Now in this picture, what are the things I am showing? What is the red color line?
Plasma membrane.
And what is outside the plasma membrane?
Primary cell wall.
Primary cell wall. Clear? In the primary cell wall, there will be cellulose, hemicellulose, and pectin only. Understood?
Yes, sir.
Yes, sir.
Now, if you take this arrangement, you will see in all plant cells. In all plant cells, you will definitely see a primary, what is it? Cell wall. In some mature plant cells, I'm confining it, some mature, what is it?
Plant cells.
Plant cells, right? Another cell wall will be secreted. Okay, in that case, say this is the plant cell. Now it is secreted the thicker plant cell wall called what type of cell wall? What type of cell wall? It is the secondary cell wall. Then primary cell wall. Primary cell wall will be pushed outwards. Primary cell wall will be here. Understood?
Yes, sir.
Yes.
So, what are the components available? If you take now, you know, black one is primary cell wall, red one is what is it?
Plasma membrane.
Plasma membrane, and here what is present is
Secondary
cell wall. In this one, there will be cellulose plus mostly it will have home lignin. Sometimes it will have home
suberin.
suberin. So What is the plant cell wall you will notice in all the plant cells?
Primary cell wall.
Primary. Some plant cells only make
Second.
secondary cell wall, correct?
Yes, sir.
In that case, the secondary cell wall has cellulose mixed with lignin or suberin. Are you clear?
Yes, sir.
Yes. Could you please take it down? What is the heading? Plant cell walls. Clear?
Clear, sir.
Use different colors, show different thickness. That is very important. You can see which one I have drawn as a thicker one.
Secondary cell wall.
The secondary
Cell
cell wall.
Done, sir.
Have you, if you complete, tell me.
Completed, sir.
Right.
Done.
Now, another one more thing. In any plant cell, now say there is a tissue, a plant in a tissue. If you take a plant tissue, in a plant tissue, many plant cell. Now this is the I am drawing in black color. If I am drawing in the black color, what is the meaning? Which one I am drawing?
Primary cell wall.
Primary cell wall. Okay. So here I'm taking an equal size one. Here two primaries, two plant cells are there. Between now when these plant cells come together, these plant cells will be attached together by a material called middle lamella. What is the name of this material?
Middle
Middle
lemon.
Okay, one cell length. Definitely primary cell wall and primary cell wall only will be attached. You know, if secondary cell wall is there, where it will be?
Inside primary cell.
That is so this it's inside, no. So all the time, primary cell wall and primary cell wall are, what is it?
Connected.
Primary cell wall and primary cell wall are attached, and this is called by the name, what is it? Middle lamella. Okay, in young tissues, it is made of the material called, what is it? Pectin. In mature tissues, it is made of a material called calcium or magnesium. What is the material?
Pectate. Calcium.
Calcium pectate or magnesium pectate. Okay. So, could you please mark it down? Mahal, நான் ஒரு 4 minutes-ல வந்துருவேன். I'll pray and come. நான் இன்னும் அசர தொலைவில். Okay. I'll pray and come. Please wait. Okay.
Okay.
Jazakallah khair. Thank you for waiting. So, are you clear regarding the middle lamella?
Yes.
Are you clear regarding the middle lamella? Right. So, middle lamella is a glue-like material. It is gluing, pasting the two adjacent primary cell walls. Okay. So, if you compare primary cell wall and secondary cell wall, which one is thicker?
Secondary cell wall.
Secondary cell wall is thicker. So which one could be more flexible?
Primary cell wall.
Primary cell wall is comparatively flexible and primary cell wall is permeable. Permeable means any material can go through the holes in the primary cell wall. But when you come to the secondary cell wall, suberin, lignin-like materials are fully sealing the spaces. So can any material move through the secondary cell wall?
No.
No. So secondary cell wall will be hard, secondary cell wall will be rigid, secondary cell wall is not permeable to any material, secondary cell wall is thicker. So it is totally giving a protection like thing. Understood? Are
Yes, sir.
you clear?
Yes.
Yes. One more thing, in the cell wall, if you take the cell wall, in the cell wall, in some places, now here see, I'm maintaining the structures. If I am drawing in the red color, what is it?
Plasma membrane.
Plasma membrane. Okay, if I am drawing in the blue color,
Secondary cell wall.
secondary cell wall. Now I am drawing what is it? Secondary cell wall. In some instances, there will be spaces like this. Can you see this space?
Yes, sir.
In that space, not only secondary cell wall, even primary cell wall will not deposit. Just I'm drawing one, there can be many, but no need to draw many, right? Understanding only the diagram, no? These kind of places are called by the name. What is the name given?
Pit.
The pit. Okay. In the pit area, in the pit, will there be primary cell wall? Will there be secondary cell wall?
No.
No. There
No,
can
sir.
be many pits there, but just I have drawn one pit for your understanding. Please mark it down, the pit. Okay, have you marked it?
Yes, sir.
Yes. So
Yes.
now take the notes. Please go through this cell wall notes. Okay. Anything you don't understand me, just indicate that point to me. I will explain you. Don't miss anything without understanding. Anything, even a small point, if in a single word, if you don't understand, please tell me.
Sir,
Yeah, ma'am.
sir, they have said water can move freely through free spaces of cell wall. In this, did they mean free?
Primary.
Did they mean free spaces for the pit?
No. If you take the primary cell wall, okay. If you take the, now normally, if you see the cell wall, okay. If you take the cell wall, the cell wall, what is present? Cellulose fibrils are present in the cell wall. There are, what is it? A fiber arrangement is present. Something like this is now. Can you see? Can you see?
Yeah, sir.
Yeah. So here, can you see there are spaces? Anyway, now these are some chain line. If you put water on it, will the water go to the other side? Tell me.
Yes, sir.
Yes,
Yes.
definitely. Because there are spaces. It's like this. Can you see? This is the TEM diagram. Can you see the spaces?
Yes, sir.
So is it compulsory for the water to move through the pits?
No.
No, I don't tell the water will not move through the pits. That's not the meaning. Understood?
Yes, sir.
Definitely water can move through the, what is it? Space. But what these people mean it is, in the cell wall, this is a network like cellulose fibers are making a network. In that network, there are spaces, correct?
Yes, sir.
Through those spaces, materials can, anyone that's fit to the size can move, correct?
Yes.
In the secondary cell wall, all these holes are filled by lignin or suberin. Then what will happen?
No water flow.
No, nothing can move, correct? Yes or no?
Yes, sir.
So which
Yes, sir.
one is permeable? Which one is permeable? Now water can move freely through the spaces of the wall mean? Yes, that may
Primary
be
cell.
indicating the primary cell wall. Anything else you can ask, no problem. You just point out, sir, what does this mean? I will tell you.
So what do you mean by lignin cements anchor cellular fibers together?
Yeah, I told you here what is there. Here what is there? The cellulose fibers are there, right?
Yes, sir.
Yeah. So it is almost like a concrete. When they, have you seen how do they make a concrete? How do they make a concrete? First of all, they will develop a wire mesh like this. Correct? Have you seen?
Yes, sir.
So they should be tightly hold together. That tightly holding to them together is the one done by the cement poured on it. Correct?
Yes, sir.
So because of cement, those metal bars are tightly hold together. It is making a harder structure. So in this diagram, if this is the primary cell wall, okay, on this, don't leave lignin. On this, if you put cement, what will happen? All these fibers will be
Get hard.
tightly fixed together and it will be becoming a full hard structure, correct?
Yes, sir.
So point it is cement, that lignin is a cement. Simply connect it. Lignin is a, what is it? Cement. What does cement does in concrete? Same thing lignin does here. It's an anchoring work. Holding, allowing the fibers to tightly hold on the cement. Now they are given extra strength, correct?
Yes, sir.
That one. Are you clear?
Yes, sir.
Yeah. Anything else if you want, you can ask me. No problem. Anything you want, you can point out. I will explain. in this one, the last point tells you about the what is it? Plasmodesmata. Okay. If you take plasmodesmata, here see this diagram. Can you see this diagram?
Yes, sir.
Yes. This is the pit region. Which one? Which region? Pit region. This is one cell's cytoplasm. This is another cell's, what is it? Cytoplasm. These two cytoplasms are connected by a jelly-like material. That jelly-like material connecting the two cytoplasms is called by the name, what is it?
Plasmodesmata.
Plasmodesmata. Through this is a bridge-like, materials in the cell 1 can go into cell 2. Materials in the cell 2 can go into cell 1 through this plasmodesmata. Understood? Regarding that also, a small point is given to you. Right. Check it out. Anything you want to clarify, don't miss any point. If things are okay, tell me, I will go to this function.
Okay.
Okay.
Okay, right. Now, if you take the cell wall normally, its normal function is protection and support. In our body, it is done by whom? Skeleton. Okay. So, cell wall does the function of a, what is it? Skeleton. So that's why in plant cells, in not only plants, in plant cells, cytoskeleton's need is high or low? Low. Why? Because cell wall is there. It is giving protection and support. Then cytoskeletons need to give support is comparatively low. But in an animal cell wall is absent, mean okay, then the support needed is very, very, very
high.
Understood? Understood? No need to further explain it, that point, no?
No need.
Second one, I already told you regarding turgidity, correct?
Yes.
Yeah. In developing turgidity, you know, very importantly, vacuole plays a role, correct?
Yes.
Yeah. Same time, with the support, okay, cell wall is also important to develop the turgidity. Okay, when the vacuole enlarges, it will go and press the cell wall. When cell wall is hard now, it will tightly reject the pressing. Yes or no?
Yes, sir.
Did you understand what I told you? Here, the cell wall say, here, a vacuole say, When the vacuole is enlarging, it is taking water, becoming large, large, large mean, okay, it is going to press the whom?
Cell wall.
Cell wall. When it presses the cell wall, that means it applies a force on the, what is it? Cell wall. As a reaction, cell wall will apply an opposite force. Understood?
Yeah, sir.
That is very important to get that stiffness. Okay. Third one, because of turgidity, one benefit. Read this one.
Prevents bursting during turgidity.
Right. You know amoeba. Do you know amoeba?
Yeah, sir.
Yeah.
If you put some amoeba inside distilled water, amoeba will enlarge in one place. Dub. Amoeba will burst. Now see, there are aquatic plants there, correct?
Yeah.
Are their cells bursting like that?
No.
No. Who is helping?
Cell wall.
Cell wall is helping one side vacuole is helping. That's true. It is osmoregulation, correct?
Correct.
So one side cell wall is helping, one side vacuole is helping. Another one, it is limiting and controlling the, what is it? Cell growth. Okay. If you, I used to take, remember, when I am explaining things, I used to take these types of examples. Okay. Okay, don't mistaken me, I'm choosing this. Okay, Japanese feet. Okay, binding. Japanese, what is it? Feet binding. See this feet. What has happened to this feet? Can you see her finger here?
Yes.
What the thing they did was, in Japan, once upon a time, there was a big belief females with small feet will bring fortune to the family. So this kind of shoes were the only choices given to the girls. Understood?
Yes.
So because of this, what has happened to their feet growth? Can you see? So a harder material is limiting and controlling the growth. Can you understand the way I am connecting it?
Yes.
Read the fifth one. Read it. Read the fifth one.
Component of
Components of apoplast pathway. Right? In the plants, okay, there are several methods of water movement. There are several methods of, what is it? Water movement. 3 routes are there. Unit number 4 you will study. In that route, okay, there is one route called apoplast route. Another route is they are called symplast route. What is the name of the routes? What is the name of the route? Okay. If you take the apoplast route, it will go from one cell wall through the spaces, will go to the next cell wall. Through the spaces, it will go to the next cell wall. That kind of route is what kind of route? Apoplast route. One cell cytoplasm going to the next cell cytoplasm through the plasmodesmata. Then from that one going to the next cell cytoplasm through plasmodesmata. Like that, if it is going, it is symplast route. So what are the two types of route pointed here? What are the two types of route? Just read it.
Apoplast and symplast.
Apoplast route and symplast route. Okay. Cell wall spaces. Cell wall spaces. Not going through the cytoplasm. Which route it is? Which route it is? Apoplast. From one cytoplasm through the plasmodesmata going to another cytoplasm. From there through the plasmodesmata going to another cytoplasm. That is what kind of route?
Symplast.
Symplast route. Cell wall is contributing in which route? Which route?
Apoplast.
Apoplast route. As a skeleton, it is maintaining the shape. As a skeleton, it is helping the plant to stay upright against the gravity. We are standing upright against the gravity, correct? Because we have a skeleton. Am I right?
Yes.
Yes.
So most of these works are skeletonous works. This is because it is a skeleton, right? Am I right? This is also because it is a skeleton, right?
Yes.
This is also because it is a skeleton. This is also because it's a skeleton. Hard. This is also because it is a skeleton. This is also it is because of what is it? Skeleton. Only that apoplastic pathway part is non-skeletal function. Clear? Are
Yes.
you clear? Right. So another small portion only they are to complete the cell biology portion. Only we have to study the cell junctions and what is it? Plasmodesmata, extracellular matrix, ECM. ECM we have to study. With that one, this tute will be finished. Cell cycle tute I will send you. You children take the printouts. Okay, you take the printouts and stay ready. Soon we finish this one. We can start the discussion on what is it? Cell cycle. Right? Same time, I have told you to get ready for a chemical basis of life.